We now turn to the production of proteins, or gene expression, and I want to provide an overview for how cells take the sequence of DNA and convert it by transcription and translation to a sequence of amino acids, which would then make up a protein. Gene expression is the production of a functional gene product. And the first stage of gene expression is transcription, the synthesis of a single-stranded RNA molecule based on base pairing with a single-stranded DNA template. Now there are some functional gene products that function as RNA, but protein-coding genes go through a second step of gene expression, which is translation. And translation is the production of a polypeptide based on the RNA nucleotide sequence. There are three major types of RNA. An mRNA, or messenger RNA, are the type that actually get translated because they encode a polypeptide. The other two types of RNA function as RNA molecules and do not get translated. Transfer RNA, or tRNA, carries amino acids to the ribosome, and the ribosome is the structure that actually translates mRNA to create the polypeptide. rRNA, ribosomal RNA, is part of the ribosome's structure, and it helps the ribosome to translate RNA into a polypeptide. Again, both of these types are not translated, but they are transcribed from genes that encode those sequences. The process of gene expression then involves transcription, and the RNAs, rRNA and tRNA, are transcribed to make RNA, and then they will end up leaving the nucleus to go do their job. mRNA, by transcription, encodes a polypeptide. This is the type that also leaves the nucleus to be translated by the ribosome. Translation will result in a polypeptide, which folds and becomes modified to become a functional protein. First, let me talk about what it is that we find encoded along the length of chromosomes. Remember that we have 22 pairs of autosomes and 1 pair of sex chromosomes, and along the length of these chromosomes are about 23,000 protein-coding genes. 21,000 is a conservative estimate. The max that I've seen predicted is about 23,000 protein-coding genes. If you look at the individual chromosomes, chromosome 1, which is the largest chromosome, with 249 million base pairs, this contains 2,100 genes. So along the length of this double-stranded DNA molecule, there are sections, and each section encodes a protein-coding gene. Chromosome 22, which is the smallest, 51 million base pairs, encodes about 600 genes. That just gives you an idea of how many genes are found along the length of these chromosomes. Now, in addition to protein-coding genes, we also find tRNA and rRNA genes, which are transcribed to produce those two types of RNA. A long double-stranded DNA chromosome has along its length genes. Genes are interrupted by non-coding regions, but if we zoom in on a gene, remember that a gene is made up of double-stranded DNA. One of those two strands of DNA, the same strand every time a protein is made, is going to be read through the process of transcription to create an RNA molecule that is made based on base pairing with that template. Then the RNA molecule is going to be read in a series of 3 nucleotides called codons, and the structure that reads that with the help of rRNA and tRNA is the ribosome. The ribosome will read these 3 nucleotides, and for every 3 nucleotides, incorporate a specific amino acid based on the sequence of the 3 nucleotides. Transcription results in the RNA based on a DNA template, and translation results in the polypeptide based on the sequence of codons. The codons have specific amino acids in which they encode. Every possible combination of 3 nucleotides is shown here on the genetic code. CUG, for example, encodes the amino acid leucine. In this genetic code, all the amino acids are given in their 3-letter abbreviation. Some genetic codes that you might find have the entire name of the amino acid written out. Some have just the single letter abbreviation. This one has the 3-letter abbreviation, and this is the most common form. I want you to notice that most amino acids are encoded by more than one codon, some 6, some just 2, and most by 4 codons, those 4 codons differing at the 3rd position of the codon, the 3rd nucleotide. In addition, there is a start translation codon and a stop translation codon. When these are encountered in the mRNA, the ribosome knows to start and to stop translation. These are not start and stop translation or start and stop codons for transcription, only for translation. The code is almost universal. Almost every living organism uses the same code, including bacteria. So from bacteria to humans, all living things, with a few exceptions, use the same genetic code in translating an mRNA to create a polypeptide. Now, in double-stranded DNA, remember we've got 2 strands that are base paired with each other. Only one of these 2 is actually going to be used as a template for transcription. That's the template strand. The other strand is called the coding strand, and notice that the coding strand looks exactly like the RNA that's produced, except that the coding strand has T's because it's DNA, and the RNA has U's because it's RNA. The resulting RNA is read from one direction to the next, or to the end, and every 3 nucleotides in the codon encodes an amino acid. AUG, in the genetic code, encodes methionine. That's also the start codon, and so all polypeptides start with methionine. The stop codons are also found in the RNA, and a stop codon does not encode an amino acid. It just tells the ribosome that it's done. The last amino acid that was incorporated in is the last amino acid in the polypeptide.